So I have two polysaccharides for us to compare today. And the interesting thing about these polysaccharides is that they are only different in one very small way. But this difference is actually really important in the structure-function relationship. We'll start with cellulose. So cellulose is a long string of glucose molecules that are linked together in a beta 1-4 glycosidic bond. Humans are not able to digest cellulose. It is just like fiber, right? Fiber goes right through us. Cellulose is super interesting because it's very structural, very rigid. So if you imagine all of these glucose molecules, we know glucose molecules and carbs in general have a lot of hydroxyl groups. We have all these opportunities for hydrogen bonding. Well, cellulose's structure is much like the ice form, the solid form of water, in that those hydroxyl groups have really maximized the number of hydrogen bonds they can make. The structure, the lattice, is such that we're just making all these beautiful, perfect hydrogen bonds. Hydrogen bonding happens within the single strand of cellulose, between two different strands that are next to each other, and even between these kind of sheets of cellulose fibers. That makes cellulose exceptionally strong because of the number of hydrogen bonds, and actually, even though you'd expect it to be water soluble because it's so polar, cellulose is water insoluble because it's already perfected the number of hydrogen bonds. So breaking those hydrogen bonds to then interact with water would actually result in fewer hydrogen bonds, which is interesting. I want us to compare cellulose that has a beta 1-4 glycosidic bond to its cousin starch, or amylose. The only difference, the only substantive difference between cellulose and starch is that starch has an alpha 1-4 bond. It still is a massively long string of glucose molecules, but the glycosidic bond is not beta, it's alpha. And so this leads to irregular packing and a helical shape. And because there's an alpha 1-4 bond, humans can metabolize it. We can break starch down. Thank goodness, because fries and potatoes are amazing. Let's wrap things up, folks. Just a quick side note, right? I mentioned that sugars undergo additional reactions that we're not discussing in this class. We can add sugars to proteins. This is called glycosylation, and this is a focus of Carolyn Bertozzi. We can covalently modify proteins at asparagine, serine, or threonine, and this can have a wide variety of functions. One of the functions that you might be familiar with is blood type. So the ABO blood type is actually dependent on the type and arrangement of sugars displayed on your cell surface. So the O blood type, for example, has the simplest type of sugar attachment. Compare that to the A, and you'll see that A has an additional type of sugar on there. Compare that to the B. B also has an additional sugar compared to O, but just different than A. That wraps up carbs, folks. A brief introduction to carbohydrates, structure, and function. We learned about how diverse carbohydrates are. They have both linear and cyclic forms that we should be able to look at. And then we really focused in on the anomeric carbon. Is it alpha? Is it beta? What does the glycosidic bond look like? I will leave you with your cat meme of the day. Thanks, everyone.